All right, hello there. My name is Cecilia. I am a senior in the Chemical and Biological Engineering Department, and I'm here to present today on optical nanosensors and deploying them into capillary culture systems as a means of quantifying an oxygen gradient within these cultures. So before we get into anything, what exactly are nanosensors? And they're nanoparticles that we engineer. They're about 150 to 200 nanometers in diameter. The biggest thing about these is that they're selectively and reversibly able to bind and recognize an analyte of interest to have enough selectivity at very narrow ranges that we can measure the concentrations of these analytes. And so the way that this works is that binding induces a change in the optical properties of this molecule, and that change we can measure usually by a plate reader and then correlate that change in fluorescence to the concentration of whatever we want to study. And there you can see a picture of some of our nanosensors. They're often very colorful and very pretty, and they will fluoresce under UV light. So it's also very fun to work with just in general because it is so colorful, and we can make optical sensors of all the colors of the rainbow. They are made of three major components, the first one being the hydrophobic core. This is the thing that mostly promotes the selectivity. It filters what particles are able to even enter the sensor itself contained within the core. But then because that the sensor itself is very hydrophobic, it does protect that sensor from the outside environment. And then you have recognition, which is the elements that bind to whatever analyte we're interested in. In this case, for the oxygen sensors, we have a signal dye, which is PTFT-PP. It's the thing that actually binds to the oxygen and changes its fluorescence. But then we have a reference dye. The reference dye is unreactive to the analyte of interest, and its fluorescence signal does not change. So therefore, we can always quantify how many sensors are in that environment at a given time, even when the signal of the other dye is quenched. And then finally, we have the PEG lipid coat. It's biocompatible. We can deploy it in our capillary bed systems. And most importantly, it makes the sensors stable in an aqueous environment, which are most biological environments. So when we're looking at the oxygen sensor itself, it uses a quenching mechanism, as I mentioned previously. Basically, the oxygen binding to our signal dye turns off or quenches our fluorescent signal. And that's the thing that we can measure and figure out how much oxygen is in that particular region based on how much fluorescence decreases over time. And so basically how we do this and why we're able to do this most importantly is because the signal dye reaction occurs at equilibrium with the analyte concentration in the environment. Therefore, the signal quenching is directly proportional to, inversely proportional to how much oxygen is in the environment. Again, the reference dye is unreactive, and it's the constant fluorescence which enables us to quantify how much sensors are actually in that region. And then here's just an overview of what that mechanism looks like. Essentially, our oxygen enters into our polymer coat, selectively filters, it binds to our signal dye, quench the unreactive reference dye remains, and then we can measure both of those, the fluorescent ranges, the emission and excitation in those ranges, and get our results. Here is the mechanism specifically. As I said, light will come in, it'll excite both dyes, both of them are will fluoresce in the red range. And we have a really nice bright red emission without any oxygen present. But then as oxygen comes into our sensor and binds to our platinum dye, also we see a decrease in that red signal, although it is a little bit still red because that dye is still fluorescing in that range. But we do see a change in emission that we can quantify. All right, and then we're looking at the dyes themselves. Again, quenching mechanism. We see here how platinum has that really nice aromatic ring structure to select or cooperatively bind to oxygen, while dye not many opportunities for oxygen to bind, does not bind oxygen at all. And then here's some evidence from one of our previous alum at the Cash Lab describing how that inverse proportional equilibrium occurs. So as you can see, this graph shows that the signal dye, platinum dye, basically its signal decreases as oxygen concentration reaches atmospheric levels, which is that black line at the bottom. Our 0% oxygen is in the blue. And then when we're looking at the capillary bed specifically, which is the culture system which we want to deploy these sensors into, this is a technique based on the Winograd-Seq column. The Winograd-Seq column facilitates the growth of diverse microbial populations through a progressive aerobic and anaerobic zone that's meant to mimic the conditions that we find in ecological conditions, like in sediment and water. As you can see in this diagram here, we actually move up through a bunch of different metabolic layers. So microbes at the bottom will primarily be using sulfur, and we move up towards lactate, for example, or iron towards the middle, and then towards the top is where aerobic and where oxygen is the main metabolite being used. And so we really want, the whole goal of this project is to be able to deploy 4 different types of sensors for these 4 different analytes I previously mentioned throughout the entire length of the column, but do it on the microscale, which is where the capillary beds come in. We want to make a Winograd column and see if we can make this gradient of metabolites on a small scale without having to build a massive column. And the way that we're going to study this is through the use of Pseudomonas aeruginosa. They're facultative anaerobe. They're particularly well suited for this study because they're facultative. They can still thrive and proliferate in oxygen depleted environments, so we don't have any cell death along the length of the tube if an oxygen gradient is established. What's really cool is without even having to deploy our nanosensors, we actually see that these bacteria fluoresce under UV light towards the top of the tube. And so we're thinking that aerobic respiration is likely responsible for this fluorescence, but the fact that this fluorescence changes across the length of the tube suggests that our bacteria are actually utilizing different forms of metabolism along the length of the tube itself. We don't know if necessarily if it's oxygen just from this picture alone, but it's really, really promising news. And so when we finally integrate the sensors, we do actually see an establishment of the oxygen gradient. It's really, really cool. And the fact that we can deploy these systems and measure them across time, meaning we can come in and take a measurement at 12 hours, at 24 hours, and even upwards to 48 to 36 hours without even seeing any significant cell death is really amazing. So we can actually see the development and how this oxygen gradient progresses and how our bacteria are responding to slowly depleting oxygen conditions before cell death occurs. And it's really important that I want to mention how this is precisely measured. We actually used a modified 6-well plate, and we standardized all of our capillary tubes using an optical density reading at 600 nanometers to just make sure that there's roughly the same amount of cells in each of our capillary tubes. Because it is such a small volume, it is a little hard to standardize, and we're working on improving those techniques to make sure that we have roughly the same amount of cell count in every single tube so we can replicate this across multiple experiments. But again, potential for longitudinal study, we can look at this and how these cultures develop over a span of 1 to 3 days and see if these bacteria respond any differently to changing conditions. And then here is that gradient quantified. This is the average fluorescence of our platinum dye, excitation 540, emission 650 nanometers. I know the axis looks a little funny. It's because the axis corresponds to the orientation of the open tube that I showed in those previous pictures. So the open tube was on the right, hence why our pictures here, our axis here is on the right instead of on the left, because that's where it's open to air. That's position 0, and then we see the progression. And again, because platinum dye inverse is the fluorescence is inversely proportional to the presence of oxygen, we see it really fluoresce at the bottom of the tube where there isn't any oxygen, but no fluorescence at all at the top. Please note that there is a large deviation at oxygen-deprived regions. I'm going to get into that on our next slide. And that's really what the challenge is going forward when it comes to these experiments, is that because we're using a modified 6-well plate, where basically we just cut little, using a serrated blade into the circumference of the 6-well plate and place them vertically across. We do see some deviation in how the plate reader can measure these capillary tubes. As you can see in column 2 really emphasizes this, how you see how 2 of the 3 measurements that were taken on the vertical axis are really dark and have very nice values, but that 4th one, or the 3rd one on the right, has really light values. That's because our capillary tube was off the center of the axis itself and it didn't capture the full measurements. And our plate reader obviously can't account for the curvature of the tube, especially if it shifts as it goes into the plate reader, which is incredibly common because of the way that we cut these plates is not necessarily completely fit to the tube itself because we're doing it by hand. And another thing I really want to emphasize is the missing data in this experiment. As you see along that center line, because of our plate reader data, we actually miss about, I think it's about 2,000 microns worth of information along the vertical axis, just due to the well carrier limitations of the program that we use to take these measurements, and which explains our high variability in those oxygen deprived regions and why there might be experiment disparity, because each measurement that we take might not necessarily has a large degree of error. And we have a bunch of variability due to cell count across the different capillary systems that we're hoping to standardize. And then here's a picture of the modified plate itself. And then I highlighted our capillaries in red. And as you can see, it's a little, I thought it was really brilliant the way that we were developing this, because we didn't really have any other way besides using a confocal microscope, which we don't have on hand at the lab right now. And that also requires curvature adjustments in order to quantify the tubes. But as we're looking into using a liquid culture when we're first inoculating the Pseudomonas aeruginosa, that really improved our replicability for cell count as it enabled us to better distribute those cultures within our liquid broth and nanosensor solution. And then we're also currently working with Biotech, which is the company that developed the Gen 9 software, which we use for our plate reader. So we can produce a program specific to our lab that overcomes those well capacity limits, so we can get that missing data incorporated into our sensor design and our results. And then kind of looking forward and like why this project is so promising and amazing is that we have not just applications in the lab, but applications in biotechnology moving forward. But looking at the small scale, our first phase of this project was really saying, do these capillary systems even establish the oxygen gradient that we see in our Winogradsky and our other larger column systems? The answer is absolutely yes. Right now, we're looking into seeing if these systems can establish gradients for other metabolites, such as iron, sulfate, and lactate. Right now, I'm working on the iron sensors project in order to like really tune in and optimize the way that our iron sensors work and how long that they last and how they behave in different systems and like how selective are they. So then we can start looking into establishing an iron gradient in our capillaries and then studying the use of our iron sensors in that regard. And then looking long-term, way past my time in this lab, can these sensors be deployed to quantify the speed at which microbial systems, like diverse microbial systems, environmental samples, medical samples, reach a steady metabolic state? And can we deploy these 4 different types of sensors concurrently to study the development of a metabolite gradient along the length of our capillary tube? Which is really cool if you think about it, because that has huge potential for diagnostic medicine. Due to their highly sensitive ranges and sensitivity, might I add, that is being improved every single day with new nanosensor design, we can start developing sensors that can mark for proteins or other early markers of disease, cancer metabolites, or environmental contaminants before symptoms emerge on the macroscopic scale. So then we can implement earlier intervention and hopefully more improved treatment for these patients. The next major section where this can be applied is in ecology. As previously mentioned, we can take environmental samples such as those from like Clear Creek, and we can put those in our capillary beds. And if we can quantify how many cells are in there, then we can start deploying our sensors in and quantifying how natural systems with all of the millions of different types of species that are in there will respond to pollutants, soil changes, and other effects of climate change that we can really start to study on the microscopic scale, which is really cool. And then finally, which is in regards to pathology, we're really looking into, our lab is looking into the development of biofilms. Biofilms are structures produced by antibiotic resistant bacteria. They're very like thick and viscous, and it's very hard for any type of treatment to penetrate through the film, which is what makes the bacteria very difficult to treat. But now being able to deploy these sensors, now we can start studying how the composition and the structure of those biofilms change across time. And if we can like incorporate that data and figure out like the structure of the biofilm in a dynamic environment, we can actually start developing like drug treatments or other ways that we can more effectively treat like antibiotic resistant bacteria. So in conclusion, the capillary bed model is a very promising system for the study of metabolic progression, both in biological systems and ecological systems on the micro scale. However, we still have a lot of work to do in terms of standardizing our imaging, incorporating diverse species so we can encapsulate a system that more closely resembles our bodies or an environment, and then ensuring that our oxygen, our iron, our lactate, and our sulfur sensors connect selectively and accurately to quantify metabolic flux in these complex systems. Perfect. So thank you so much for attending today. Again, I'm Cecilia Neppel. I work for the Cash Lab.